Today's topic is mitosis and meiosis.
My name is Shahana,
My name is Hannah,
and we'll be your hosts for today's segment.
Today we will be examining our friend Chip the germ cell.
Chip is a diploid cell that consists of 8 chromosomes and lives in the testes. In order to prepare for mitosis, Chip must undergo interphase, a part of the cell cycle which consists of 3 phases: G1 phase, S phase, and G2 phase.
The first phase is G1. This is where Chip will quickly grow. He then goes through the synthesis phase, which is where his genetic material is copied and then joined together by the centromere to form a chromosome. After all that work, Chip will then restore his energy in the G2 phase, as well as produce structures such as protein, which are needed for the next cell division.
After interphase, Chip is now ready for mitosis, which consists of 4 phases: prophase, metaphase, anaphase, and telophase. The first stage is prophase. This is where the chromatin will condense and form chromosomes. The nuclear membrane breaks down, releasing the chromosomes into the cytoplasm, and then disappears. The centrioles will then move to the opposite sides of Chip and form spindle fibers that will help move the chromosomes within Chip.
The second phase is metaphase. Spindle fibers will guide the chromosomes to the center line and then attach to the centromere of each chromosome. The sister chromatids are then pulled to opposite sides of the cell.
In anaphase, the sister chromatids are pulled apart at the centromere to opposite sides by the shortening of the spindle fibers. While some fibers shorten, other microtubules in the spindle apparatus lengthen, which forces the poles of the cell away from one another. Now, each pole contains one complete diploid set of chromosomes.
The final stage of mitosis is the telophase, where the chromatids begin to unwind and become less visible. The cells are now 2 separate cells, and the spindle fibers breaks down. The nuclear membrane reforms, and the nucleolus within each nucleus.
At the end of mitosis, 2 daughter cells called spermatocytes are produced. One replenishes the cell and the other develops into a primary spermatocyte. This primary spermatocyte then undergoes meiosis. There are 2 types of meiosis that Chip will go through. First, meiosis 1, then meiosis 2. The first stage of meiosis 1 is prophase 1, where synapsis, the lining up of 4 sister chromatids in a tetrad, occurs. Here, the genetic coding is shared and exchanged between the homologous pair, known as crossing over.
The spindle fibers then attach to the centromeres of each homologous chromosome, lining them up along the center line. The spindle fibers shorten and pull the homologous chromosomes to opposite poles of the cell. A single homologous chromosome, 2 sister chromatids, will move to each pole. In the final stage, the homologous chromosomes begin to uncoil and spindle fibers will disappear. The cytoplasm is divided, creating 2 haploid cells, and a nuclear membrane is formed around each group of homologous chromosomes.
The phases of meiosis 2 are similar to those of mitosis, except in the end, 4 gametes are produced instead of 2 cells like in mitosis. In prophase 2, the chromosomes are aligned side by side, then as homologous pairs in metaphase 2. Both anaphase 2 and telophase 2 contribute to the further division of the cell, where the separation between the cells become more distinct. At the end of this process, 4 non-identical gametes are produced. The daughter cells are still haploid, but contain single unreplicated chromosomes.
And this is what happens in mitosis and meiosis. Has successfully divided into 4 sperm cells.
I am Shahana.
I am Hannah.
And this concludes our presentation.